Welcome, everyone. My name is Kira Bradford, and I am co-PI and technical lead of the HEAL Data Stewardship Group. I want to thank you all today for joining us for the sixth installment in our Fresh Fair webinar series. Today's webinar is called Metadata 102, Empowering Search on the HEAL platform, and we'll focus on providing a HEAL study-level core metadata model that will allow HEAL researchers and the broader community to maximize the benefits of the HEAL platform. This webinar will consist of a brief presentation followed by a moderated panel discussion and then a live Q&A. If at any point you have a question, please feel free to type it in the Q&A box. Next slide, please. We are joined today by Andrea Tentner, who will lead our presentation. And Andrea has a master's in public health and a PhD and is a senior data scientist at NORC at the University of Chicago. Dr. Tentner is a biological engineer and systems biologist by training with a strong bent towards public health, intent on bringing traditionally siloed fields and data together to study, model, and test the health and health equity impacts of broad-ranging interventions on the complex interplay of social, environmental, and biological determinants of human health. Andrea has been thrilled to be part of the core team working to formulate a HEAL metadata model that describes data in a way that transcends field, drawing a broader set of data consumers to find and use the data. With that, I will pass it over to Andrea to begin her presentation.
Great, thanks Kira for that introduction, and thanks everyone for being here. Great, so today we're going to continue the Fresh Fair webinar series with a second look at metadata, and just a heads up, today we're going to specifically focus on study level metadata. So we'll address file and variable level metadata another time. So in the first metadata webinar, Metadata 101, we defined metadata, and today we'll briefly review that definition and we'll get into why it's important to consider the audience and purpose of metadata when deciding what metadata to provide. Next, we'll review the purpose and audience of the HEAL Initiative. I'll review the categories of HEAL study level metadata we developed with that purpose and audience in mind. And I'll provide some examples of how the HEAL metadata we developed can enable specific HEAL audiences to find what they need. Finally, we'll dip into how we're leveraging the power of the HEAL platform to keep the effort of investigators in providing the HEAL metadata as low as possible, while creating as much value as possible for the HEAL community, including HEAL investigators. We'll then have an open Q&A with myself, Alex Waldrop of RTI, and Anthony Juni of NIH, and finish with a wrap up and final words from Kira. So let's review the definition of metadata. Metadata is any information that describes your data. And study level metadata, the kind of metadata we're focusing on today, specifically will be information that describes your HEAL research study. And especially with the purpose of making the data, or again, in this case, your HEAL research study, findable and usable to people who didn't do the original data collection or study design. But you can really describe anything, including data or research study, in many ways. So there's no single right way to describe something, just depends on your purpose in describing it. So to whom are you describing the research study? What do they care about? What do they want to know or do? If we think about these questions, we'll probably hit on a type and level of description that's most useful. So we've introduced this idea that we need to provide different descriptive metadata for different purposes and for different audiences, but what does that look like? At the last webinar on metadata, John Cheadle shared an everyday example of metadata. So the metadata available on websites like Redfin or Zillow that describes houses. And some of the metadata that John called out at the last webinar were dimensions like house price, number of beds and baths, square feet. And these sites are often used by prospective home buyers. And these particular metadata may be the most useful way to describe the houses for a prospective home buyer. But there are, of course, other ways to describe houses. And there are audiences other than home buyers who would benefit from a different type of description. So for example, a historian looking to track the trajectory of different types of architecture in different neighborhoods in the city might most value a description of the architectural style of the home, as well as a tag for the neighborhood the home is in. A politician looking to understand the impact of zoning laws on new construction of single family versus higher capacity buildings might value a tag for single versus multifamily and a date of construction. And a researcher studying gentrification might value historical data on dates and prices of sale, as well as assessment and tax values and income of residents in the home census block. So the most useful description of research studies and data is one that considers the audience and purpose of the description. And in thinking of what metadata we need from HEAL researchers to make their studies findable and usable, we thought about the HEAL Initiative's purpose and audience and developed the metadata with these in mind. Human opioid use and pain conditions are intimately intertwined, and the HEAL Initiative really lives right at this intersection. So the HEAL Initiative purpose is to stimulate and draw together a vast array of knowledge being generated from HEAL-funded research and practice activities spanning fields of opioid use and pain research and stages and types of research from preclinical to clinical, epidemiology, industry, and policy. And really with the goal of fostering collaboration and breaking down silos between the study of human pain and opioid conditions and revealing the potential for harmonization across previously separate fields and stages of research. And importantly, the HEAL Initiative purpose in this is specifically translational. So to find, and not just find, but drive and involve and inform a broad community about solutions to the human conditions of opioid use and pain. So even when a HEAL-funded study is as basic science as it gets, and may seem removed from treating these human conditions. So for example, something like a mouse osteoarthritis model study of opioid receptor mechanism, the translational purpose of the study can still be stated as either one, learning more about a human pain or opioid use condition, or two, investigating a treatment, intervention, or solution to a human pain or opioid use condition. And the HEAL Initiative audience is inclusive. So from HEAL researchers like yourselves, working across a huge number of stages of research and research fields, to clinicians, patients, and other people in communities affected by opioids and pain, to researchers and business developers in pharma and industry, journalists, politicians and policymakers, and public health officials. The HEAL Initiative really is trying to bring this wide range of audiences together because it's clear that the confluence of human opioid and pain problems is not something that will be purely solved in the clinic or the courts or the community, but really requires knowledge sharing and collaboration across these silos to drive real change. So the HEAL platform will provide the main gateway and interface for this collaboration and cross-pollination and solution finding. And HEAL Initiative metadata is metadata that the HEAL platform can use to power search and discovery tools that let the HEAL Initiative audience achieve the HEAL Initiative purpose. And to that end, we've developed what we're calling the HEAL study level core metadata fields or model, with metadata elements grouped across 12 categories. And the 12 metadata categories are listed here. We expect that more technical and research audiences will be the primary consumers of metadata in these highlighted categories on the left. And the remaining categories highlighted here on the right will be especially helpful for HEAL platform visitors and consumers who may be a bit more data naive, though we also expect that these will be useful to savvy researchers. So let's run through an example use case to demonstrate some of the metadata fields encompassed by these categories and how they might be experienced in the platform interface and used by one member category of the HEAL audience. So here we'll focus on a patient or family member impacted by an opioid use or pain condition and see how they'll use metadata to find what they're looking for. So here's a hypothetical. I'm a patient with a pain condition. I'm a cis woman in my mid-30s. I'm seeing a doctor on a standard of care treatment plan that includes opioids. It's working okay, but not great. I still have some pain. And I and my family are a little bit worried about the possibility of becoming addicted to opioids if this is the long-term treatment plan. And I want to understand what other treatments are out there or being newly developed for my conditions that I can talk to my doctor about them. And I head to the HEAL platform because I know that's the place to go to learn about research on pain and opioid conditions. I see that there are 613 studies, and there are a bunch of filters I can use to filter down to studies I'm particularly interested in. The filters all provide drop-down menus, and you can see the drop-down menu expanded here for the treatment study stage search category. These drop-downs guide me in the dimensions that I can search on, so I don't have to rely on knowing the right terms to use in a free text search. And importantly, each of these filters available to the user represents a HEAL metadata field, where that metadata has been provided for each HEAL study by HEAL investigators. And behind the scenes, each of the 613 studies have provided the HEAL metadata for their study to power the filters the user sees. And here we're showing those investigator-provided values for 4 example HEAL studies. So I'm looking for a treatment. So I filter on studies with a treatment focus. And this filters out study 4 over here, which is focused on studying a condition, not a treatment, intervention, or solution for a condition. And I'm specifically looking for treatment for a pain condition. So I filter on studies with pain for the condition category. Study 4 has a condition category of opioid exposure, so it would get filtered out here even if it hadn't already been filtered in the last search category, and the other 3 still remain. And I'm looking for studies that are either trying to discover new treatments or studies that are accumulating evidence about efficacy of already discovered treatments. So I look for studies at the treatment discovery or treatment efficacy treatment study stage. And this filters out study 1, which is looking at treatment mechanism, as well as study 4, which we already know is not studying a treatment at all, leaving me with just study 2 and 3 still standing. And as a patient taking opioids for my pain condition, who's not finding full relief and who's concerned about becoming addicted to opioids, these are likely really interesting and exciting studies for me to find. So study 2 looks like it's investigating novel and specifically non-opioid drugs for the treatment of pain conditions. And you can see from the age apply and gender apply metadata fields that the drugs are being developed or tested with an adult population in mind, neutral to gender. And this study might be a purely biochemical study, something like a small molecule screen looking for good candidate drugs to inhibit activity of a protein that's responsible for amplifying an initial pain stimulus in the nociception or pain perception signaling pathway. As a pain patient, I can also see that the data release date is about a year out, so I know approximately when to check back to see if there's data or findings from this study. And study 3 looks like it's investigating a novel behavioral psychotherapy intervention for the treatment of pain. You can see from the age apply and the gender apply metadata fields that the intervention is being developed or tested with adult and adolescent populations in mind, specifically for cis female gendered individuals. The study might be a pragmatic clinical trial implemented across several clinics in a health system, enrolling only cis female patients and testing the efficacy of an individual psychotherapy sessions to reduce pain perception and increase physical function. And since I'm a cis female adult patient, I know that the results of this trial will likely be relevant to me. If I were a male or other gender identity patient doing the same search on the platform, I might still be interested in following the study, but would realize that the results might not fully generalize to me. And for this study too, the data release date is a bit less than a year out. And since this is already an efficacy trial, if it turns out that this intervention is effective, it's possible this treatment could make its way to me sooner than later. And finding these studies lets me know as a patient that there is active research in the interventions for pain that might work better for me and let me avoid opioids. And I'm empowered with hope and also knowledge that I can really take into my doctor's office to better advocate for myself. And I'll just flash past them now, but they'll be available in the slides PDF for you to review later. The use case we went through is just one example. We've fleshed out a wide range of use cases to motivate these core metadata fields. So we've seen an overview of the categories of metadata that HEAL will ask for and how specific fields within those categories really powers intuitive search on the platform and adds a lot of value for a wide range of audiences. You might be wondering, how do investigators provide this metadata and how big a lift will it be? So we know that investigators already provide required metadata via various sources. So you provide metadata to the NIH during the award application process, to clinicaltrials.gov during the study registration process if your study is a reportable clinical trial. And if the study is already registered at a data repository, possibly to that home repository. The platform works to maximize the value from existing metadata sources to also minimize additional work by investigators. And the way this works is that through a platform-mediated study registration process, the investigator or their proxy will claim the NIH application ID for their HEAL study and link their clinicaltrials.gov identifier and home repository identifier if available. The platform will harvest metadata from those sources, pre-populate that metadata to a HEAL interactive form instance, and share the form instance already partially filled out to the investigator. The investigator or their proxy will be asked to provide only the balance or supplementary metadata, so only that metadata which has not already been harvested from other sources via the form, and the platform will subsequently harvest the supplementary metadata from the form instance. All of this metadata is ingested into the HEAL platform metadata service and drives the platform intuitive search functionality. So just a few takeaways. HEAL study level metadata is data that describes HEAL studies with the broad HEAL audience in mind and with the goal of enabling the HEAL collaborative and translational purpose. And what we've presented here is HEAL study level core metadata version 1. We really welcome and will also provide venues for review and feedback from the HEAL investigator community. We really believe that everyone across the broad HEAL community will benefit from this intentional effort to describe studies for discoverability and usability. And at the same time, we are doing everything we can to leverage the HEAL platform to minimize additional effort on the part of investigators as much as possible to provide this metadata. Finally, we're going to provide support, sample metadata entry forms, support in completing the form, and feedback venues are all coming really soon. And that's all the material we have to present today. We're going to move to Q&A now, and we're really looking forward to discussing and getting your feedback.
Thank you so much, Andrea, for your wonderful presentation. So now we're going to take a behind the scenes look at how the study level core metadata model was developed for HEAL. So I'm going to invite our two other panelists to turn on their cameras. Next slide, and I'll briefly introduce them. So in addition to Andrea, we are also joined by Alex Waldrop, a bioinformatics data scientist at RTI International and a member of the HEAL Data Stewardship Group. In addition, I would like to introduce Anthony Juni, a data scientist at the NIH Office of the Director and Program Manager for the HEAL Initiative. So now we will stop screen sharing and begin the moderated panel discussion, which will then be followed by a live Q&A. So if at any time during this conversation you have a question, feel free to pop it in the Q&A box. All right, first question for our panel. What was the starting place for coming up with this core metadata list? And I think I'm going to start off with Anthony in the hot seat.
Yeah, thank you. And thank you all for making the time today and for being with us. Really looking forward to hearing from you all and your questions. So the HEAL Metadata Task Force began in July 2021 with the objective of generating a study level and file level metadata model. As you've heard, there were a few principles that drove our activities. We want to leverage existing metadata across multiple repositories within the ecosystem's distributed architecture to ensure we're lessening redundant burden on investigators, provide the information necessary to support a broad range of user-focused intuitive search needs within the HEAL data platform, and provide the metadata that can serve as the foundation for activities to enhance data interoperability and reuse across the HEAL research portfolio. And we want to release the model relatively quickly with an iterative development cycle to integrate community feedback and domain-specific needs. we began by examining existing distributed architectures, models, and standards, such as models implemented in the Australian National Data Service, the European Open Science Cloud, and even the NLM data model, as well as repository-specific models. We asked, what do we get for free, in essence, from these external repositories, such as clinicaltrials.gov, NIH Reporter, and external research data repositories. We crafted some search and collaboration use cases with a number of different focuses to make sure that we are capturing all the metadata that would be necessary to operationalize those use cases. We conducted some manual beta testing using a representative sample of HEAL studies to ensure that we could reliably map from the studies the necessary metadata. And then a number of brilliant people all conducted some consensus building exercises to adjudicate ambiguous, overlapping, and redundant fields. And then we're also finally working on mapping ontologies and control vocabularies to each individual metadata element and account for non-existing or ambiguous control vocabularies.
That is an extremely detailed answer, Anthony. Andrea or Alex, do either of you want to chime in?
I'm not even going to try to upstage that. That was, I want to go with Anthony's answer as well.
Same. I think we can wait for add-on questions to add to that.
Excellent. All right. So it sounds like there was a lot of thought that went into the study level metadata, as we would expect with this very overachieving group. So out of curiosity, let's talk a bit more in detail about this core study level metadata. So how many total fields are in the metadata model version 1? Alex?
Yeah, so there's actually about 60 fields, give or take. Sounds like a lot right off the bat, but know that almost half of these, probably 20 to 25, are going to be automatically filled by that information that we get for free, as Anthony alluded to, from clinicaltrials.gov or other repositories. So, and then the vast majority of those remaining 40 or so fields are going to be drop-down menus of ontologies, control vocabulary, things that most data managers will be able to easily answer about the data they're submitting in the studies that they're taking part in.
Okay. And how long would you estimate that it would take to complete this? Andrea?
Yeah, so we think that essentially we recommend that the sort of level of staff that you have fill this out is some experienced research staff who is really familiar with the study, including the study design and data analysis. And if they have that familiarity and also access to study documents like the proposal and protocol, we estimate that this should really not take much more than 20 or 30 minutes to provide the supplementary metadata beyond that which is harvested automatically.
All right. So Andrea, one thing that you mentioned in your presentation is that this is solely focused on study level metadata. But from John Cheadle's presentation in November, we know that there are different additional types of metadata that could be collected. So will there be additional core metadata recommendations in the future?
Absolutely. We have already started a second task force that is focused on really defining what will be the required elements as well as the recommended elements for file level and variable level metadata. And I'm not sure if I can venture to give an estimate for when we'll come out with those recommendations. I don't know if Anthony or Kira, you'd like to take that.
I mean, to be as thorough as we were, Anthony or Andrea, can you remember how long did it take from start to finish to define the core study level metadata?
We began in July of 2021, so it's been a number of different months and lots of thorough testing, but we do still expect there'll be iterative development. So I think so you all can do the math for tracing back how many months that was.
So out of curiosity, why did you all choose to prioritize the study level metadata?
I'll jump in there with just a brief vignette. Right now, there's really no way, no single location that you can go to look at what studies there are that comprise HEAL. I mean, for most people, unless you're good at doing a very in-depth NIH reporter query, there's just not a way to search over these data. We hardly even know what the portfolio looks like for the average researcher, just of what studies are being conducted. So starting with that, this platform is going to be a great way to just search at a high level for the types of studies, the types of questions that are being asked, the types of research modalities. So it's going to provide that first really 40,000 foot overview before we dive into what specific variables are being measured by each of those, or are they harmonizable across different studies?
Yeah, and I'll just add a couple of points to that. One is that out of the over 900 HEAL studies, a relatively small proportion of HEAL studies already have data collected and in a state that would be sort of ready for curation at the level that we will ask for. In addition to that, and I think that this just builds off of Alex's point, we wanted to focus on the study level metadata so that people can kind of hone quickly into a sort of group of studies that they are interested in. And then they can further dive into the data, the file and data set variable level metadata if they are interested in that. There are, of course, researchers who want to do a meta-analysis or who want to create a synthetic cohort out of multiple studies or who want to try to link data. They will need to have that very detailed information. However, as we noted, there is a really broad community that is the HEAL audience, and many of those folks will actually not be interested in necessarily going down to that level, but they'll be very interested to know what studies are going on and be able to see an overview of those studies and to see an overview of the study findings.
Thank you so much, Alex and Andrea. All right, so Anthony, you alluded to the fact that this group has been working on core study level metadata since last summer. And in my imagination, I imagine that this group wasn't always in total harmony and agreement. Can you all describe some of the disagreements or points of contention when developing this model?
Yeah, so I think given the diversity of HEAL studies, and there's over 900 funded studies now that are from preclinical to clinical to behavioral health and epidemiology focus, I think there's a lot of difficult discussions around what level is minimal, what level of detail is going to be essential to allow for a generalizable model across those. So even thinking with kind of a clinical background, do we want to leverage, say, ICD-10 codes or CPT-4 codes for cohort measuring cohort disease status or exposure status, et cetera? It's thinking through that level of detail, but then how do you generalize it to preclinical or domains that don't necessarily have control vocabularies for those sorts of criteria. So I think it was trying to make sure that we are at the highest level necessary. And there are many fascinating and really fun discussions surrounding what is too granular, what is too overly burdensome, and how do we generalize something to a domain that doesn't necessarily have some of the standards that might be applicable or needed.
Alex or Andrea, were there any other moments of passion besides trying to decide what the happy medium is between super descriptive and super minimal?
Trying to get the TMZ story on the drama of the metadata task force. No, I think that Andrew and I both did a good job representing kind of different poles of the equation, like a defense attorney and a prosecutor, where, I think she wanted a more maximalist approach and we were arguing for a more minimal approach. But ultimately we found that that sweet spot of justice, if you will, was somewhere in the middle that kind of provided a good balance.
I think one thing that was really beneficial and guided us was we wrote out a number of different search use cases that were from an investigator and researcher focus to someone, a person with lived experience and people out in the community or policy stakeholders and advocates. And really always keeping that in mind was, and keeping those search cases in mind was a very helpful way to adjudicate those sorts of disagreements and always try and keep us focused in scope of why are we gathering this, why are we building this model, and what is its actual purpose to serve. And I think that was something that was very helpful in thinking through those disagreements, or disagreements is maybe a strong word. So those nuances.
All right, so if I were to take the position of a HEAL investigator, previously HEAL stewards have recommended that investigators take a look at the recommended repository list via a repository selection quiz. So if I have chosen a repository and now I'm going to start taking a look at this study level metadata, how am I supposed to make this all work? What is that process supposed to look like? Andrea, you want to take a stab?
Sure. Yeah, so I think that you can come to this in a couple of different ways. So you might already be ready to, you already have your repository picked out and you already have your data ready to go, and you have started to register your study in a home repository, and you've started to provide some study level metadata and maybe some more detailed metadata at the repository. And in that case, as noted in the slides on the study registration process, and I believe these slides will be made available so you can go back and review them later. But the goal is that the platform is really going to pull in, it's going to leverage all of the metadata that you have already provided, both to the NIH, to clinicaltrials.gov if you register your study there, and to your home repository. It's going to pull in as much of that study level metadata that you've already provided as possible and map it to these core metadata fields. And we will ask you only to provide the balance, right, via this interactive form that will provide already pre-populated with the metadata that we harvested from these other sources. The other possibility is that you aren't ready for that, and maybe you're going to be in a repository that does not allow you to register prior to submitting your data. You're not ready to submit your data yet. You can still go through this registration process, claim your NIH application ID, give us your clinical trials registration ID, and we'll pull as much metadata as we can from there. And you can start on your form right away, even before you have chosen your repository or started to register your study there. And we will, when you're ready, you can come back and provide us that information, and we will grab that information from there at that time.
Thanks, Andrea. All right, our last question for the moderated panel before we open up to live Q&A. Anthony, what is NIH's stake in this?
Yeah, it's a very good question. I think from within the HEAL Initiative, we want to really ensure we're getting as much value out of the activities conducted by all of our investigators as possible, and sharing that data and making it available is really, really a great way to do that. I think with the metadata, the biggest stake is to make sure that we are setting up both investigators and community stakeholders up for success to ensure that they're able to find and access and reuse the data that would be of most importance for their activities. And as demonstrated through a number of different search use cases, they're very, very broad, from supporting new translational research and finding new drugs to implementing evidence-based practice and policies within the clinic and outside the clinic. So we want to make sure that we're keeping this very, very broad focus and supporting all of those impacts that could be derived from the data. also, this aligns with a number of different activities across the NIH, and particularly we're looking at the process for implementing the January 2023 data sharing policy, and we've worked and have been very, very grateful to have discussions with people that are leading that and learn lessons learned and think through our way of implementing that policy. And make sure that we're aligning with all of those expectations. We've also been very, very lucky to share and gain lessons learned from recent search and discovery workshops and RFAs coming out of the NIH Office of Data Science Strategy, and also have shared a lot of lessons learned from people that have built similar ecosystems, such as the Common Fund data ecosystem and other ecosystems across the NIH, to make sure that we're not starting from scratch and not reinventing the wheel, but really implementing best practice where it's applicable. And we've also explored other similar metadata models coming from the Common Fund ecosystem to the NLM's data model. So I think it's been a really great practice of both thinking what is of highest importance for our own initiative, but also how do we make sure that we're sharing and learning and building up success across the NIH.
Thank you so much, Anthony. All right, I will now open the floor to open and live Q&A. So if you have any questions, feel free to pop it in the QA box. And we will begin with, let's see, what is our first question? Do we have to use a specific tool, for example, REDCap, for data collection to integrate with the metadata required by the HEAL platform? Or can we add metadata after data collection directly to HEAL?
I can jump in here. So absolutely not required to collect data using any specific tool. I believe this question is perhaps a little bit more relevant to file and variable level metadata than to study level metadata. And I will say, as we said, that task force is sort of just getting going, but it is absolutely working to make sure that no one has to completely reformulate the way they collect their data, but that we can kind of meet everyone where they're at and get data to a common place.
So is it okay if they add metadata after data collection directly to HEAL?
Yes, absolutely.
Awesome. Alex or Anthony, anything you want to add to that?
I think mostly just to shout out that we're really exploring how to leverage tools to make sure that metadata that you're adding after the fact can be easily documented and shared and ingested. So, but that's a very, very important question. And also I think something that's, as Andrea mentioned, for us to keep in mind as we're working on the variable level metadata. So thank you so much for that question.
Next question. Is there a sandbox or paper forms that can walk us through the questions that need to be answered for projects where collaborative answers need to be constructed? For example, the idea of a siloed approach to completing this is not feasible because of the complexity of certain studies. Andrea or Alex, would you care to take a stab?
That's a complex question in and of itself. I do know that for any of these more judgment call type questions, the HEAL stewards are going to be there to help walk you through any of these processes. So if there are things that aren't working for you or things that you're not sure about, this is very much a living process as well. So maybe we need to expand or to modify to make those use cases more accessible. That's what we're here for. So please don't hesitate to reach out to the HEAL stewards, and we'll be happy to either help you fit in those data into the processes that we already have, or change the processes.
Yeah, and I'm not completely sure fully understand the question, so some follow-up would potentially be appreciated. But what I will say is that we already have a rough draft web-based interactive form that will be ready for release very soon, so that people can actually see what that form is going to look like. In addition to that, we will provide a how-to guide to actually how to fill this out. And what we will provide is the full form. And so this will not already, so when you actually experience this through the study registration process, of course, we'll be pulling in metadata from NIH Reporter and clinicaltrials.gov if that's relevant, and actually pre-populating that form. The form we have available very soon for you to look at is, of course, the fully empty form. And so what we'll direct you to in the how-to guide is also to which fields and which sections you might want to look at, because those are the ones that will probably ask you to manually fill out. And then, of course, as Alex said, I believe there may be a Slack channel that's going to debut soon that can ask questions. I think we haven't completely figured out what the mechanisms for, interacting with folks and providing support will be, but, there'll probably be a Slack channel, there may be office hours, there may be some more focused webinars or work sessions for people to come to if folks have questions or need help. Yeah.
Awesome. And in just a few moments, we'll pop in the metadata Slack channel link. So for those of you who are interested, you can join and ask questions. Next question. This person is curious, how would you use these metadata for qualitative data? I'm thinking Alex?
Yeah, I can jump in and take a stab. I would like, if this answer is not suffice too, I'd love to hear some more detail about that question. Because from my understanding, a qualitative study, even though the data that you're going to be collecting is not quite as standard as like a table of answers that you might get in our measurements. In terms of the experimental questions that you're asking and the design of the survey or things like that can still be, they would still fall within the realm of being describable by study level metadata. So one short answer that I have is that qualitative studies from this perspective will work exactly like quantitative studies, in that right now we're primarily interested in learning about what it is you're studying at a high level, what is your experimental approach at a high level, and then the qualitative difference versus quantitative is going to be something that we'll address more in the variable level metadata task force.
All right, so speaking of this task force, the next question is, how do individuals join the metadata work groups? i.e., how do we join the discussions of the we that you all keep referring to? Anthony?
Yeah, we of course would highly value and welcome all input. All of this began internally, and incredibly impressed by everybody that's worked on this. But I think our mission going forward is, in order to gather community feedback and do iterative releases, we're working on developing kind of a testing regimen and including people to gather their feedback in a structured way through that testing. But then also we have, shout out to the stewards, there's a collective board, which is a team of experts across the NIH space that are experts in their own domain and subject matter expertise. And we have considered spinning up a working group within the collective board to facilitate ongoing collaboration and community input. And I think that would essentially be the best space where we could facilitate gathering more of this input. But we also, there will be regular means of that if that would be the direction we would go, but then also leveraging GitHub and open spaces so that people can provide comments and commits and questions, and really would love to hear as much feedback and input as possible. So we're definitely thinking of ways to engage and gather that. So thank you so much also.
So I would recommend as a first step to join the Slack channel and indicate your interest in being a participant in a metadata task force group. I will note that this is version 1, so we anticipate that there will be multiple iterations because no one is perfect and we're not going to be able to capture all possible scenarios, which is why we need you to join and tell us how we can improve. All right, next question. So this person says, we took the data repository quiz a while back, and based on our answers to the questions, it recommended a repository. If we do determine that the recommended repository would work for us, we don't know next steps. Ideally, before we spend much team resources researching the repository, it would be nice to know that NIH would approve it to use, or at least provide us a list of ones that we could choose from. So I'm going to start off with Anthony.
Yeah, so there is a list of repositories, and I appreciate that you took that quiz. So that will be hosted on the HEAL Data Fair website soon, with all the list of repositories that that HEAL has examined as appropriate for studies within the HEAL portfolio. And thank you to everyone who's done that evaluation and that work. So that list is forthcoming, but I want to make sure I'm understanding the question. In addition to selecting a repository and the metadata that is collected, repository, we'd be able to leverage that as much as possible. But if there's additional questions about the overall workflow for selecting a repository and curating this, that is also something that we are considering and producing documentation and guidance to make sure that it's very clear how to go through that process of providing this metadata, selecting a repository and sharing your data, and walking through all of those requirements.
Thank you, Anthony. So the next question. How is the core study level metadata going to work for studies that refer to a DCC? Is the DCC going to fill this out, or are individual investigators required to fill this out?
Sorry, trying to unmute there. Yeah, so we've thought about this a little bit, and we think that this will probably vary from DCC to DCC. Some DCCs may want to take charge of this and fill this out for the studies that they encompass. And we have set up this approach so that they can interact directly with us. What we will probably need to do is to have some communication with DCCs who want to sort of own this process for all of their member studies, so that we can really get them the workflow process that we are anticipating here. It's not fully worked out, so DCCs that are interested, we would definitely like to hear from you, so that we can work out a process that works for you.
And just a reminder, we are here to meet you where you're at. So feel free to reach out with your specific questions if you feel like you're an exception, or if you're not sure what your next best step is. Next question. Andrea, so what kind of training do you provide to the users of the HEAL platform?
So there are kind of two different types of users of the platform, I will say, right? There are people who are going to be contributing studies and data, so researchers like yourselves. And then there are folks who, well, even if you are contributing, you may be using the platform in that manner. And then you may also be using the platform to discover other studies and to grab data from different distributed repositories and work with it on the platform. So there's sort of two different functionalities that the platform provides, right? As I said, the plan is to put together a comprehensive how-to guide for that first function. So how do you actually register your study? How do you get your metadata onto the platform? How do you make sure that your data is going to be queried from the repository that it's living at and be accessible on the platform, et cetera? we will have that how-to guide available shortly. And then we are also working on quite a big push on documentation of the platform functionalities in that second category. So how do I actually use the discovery and search functionality tools? How do I use the analysis tools, et cetera? And I believe there will be a platform demo at the PI meeting on April 11th, 12th?
11th and 12th, yes.
11th and 12th. So stay tuned for that to get a sneak peek at some of the search and analysis functionalities of the platform.
Nice shout out. All right, next question. I love this question because it's very future sighted. Is there a framework in place to evaluate how the study level metadata facilitate collaboration or secondary analysis? Let's put Alex in the hot seat.
Heat it up. I'm ready. This is a great question, because that is ultimately the point of this, is to increase that collaboration across those studies and lead to reusable analyses. I think that the first and foremost answer is citations. Ultimately, if people are using this the way that it's intended, there are going to be additional papers published simply because your research is going to be cited because it's findable in this ecosystem and can be reused for other purposes down the line. So some metric of citations is going to be helpful, but that's a really good thing to keep an eye on that we're going to think about going forward is how to measure and quantify that reusability.
All right, Andrea or Anthony, you want to add anything to that?
I will also just say that, yes, obviously citations, and everyone is, as a researcher, citations are a great thing to have. We do have other audiences for this data and metadata. So for example, the patient family member use case, for example, maybe someone from like a community-based organization that wants to implement some public health intervention for, opioid use in their community. And, those might not result in a paper or even in an article, but they that adds value. And I do think this is not something we have thought yet about how we can measure, but that is a good thing to think about. And I think we can put that on our list of things.
Yeah, I will say within the HEAL initiative, we're working on such a big and multifaceted problem that by thinking through questions exactly like this, we're trying to create and shift our current scientific culture to be one of openly sharing data. So I really appreciate these questions because the more we think about how my data could be used in the future for some other novel treatment or discovery, I think the more that kind of opens our brains a little bit to our own novel discoveries, perhaps. All right, next question. So this is a follow-up from the question about the DCC. So the question is, the clarification is the question was about responsibility. Ultimately, who is responsible for filling out the core study level metadata when you are working with the DCC? Would each site be required to do that, or is that coordinated by the DCC, or is it just a case-by-case basis where you work it out?
So, Anthony, you can correct me if I'm wrong here, but I will say in the end, I believe each investigator has this data sharing agreement with the NIH, right? And so in the end, it is the responsibility of the investigator. However, I think, DCCs may want to take on that responsibility. And I think one thing that we can, take onto our plate is to actively reach out to DCCs and talk to them about this issue and have them communicate with their member studies about who is going to be responsible for this process.
I completely agree. I think we would like to, from R01 investigators that may not have access to a DCC to large coordinated DCC efforts that are pulling together multiple different studies. I think that there could be some one-off specificity for each of those workflows and who would actually have the responsibility, be it even maybe an institutional librarian that might be providing support here. So I think that the testing efforts that we have in some of the next steps here, these are incredibly important questions to define, and we appreciate you raising them. So.
Thank you so much, Andrea and Anthony. All right, so we are coming up on time, but we'll ask one last question. It seems that clinicaltrials.gov has quite a bit of the meta information you're looking for. Can you search this resource for the applicable HEAL studies and compile the information from there?
Yes, absolutely. So that is certainly the plan. And, I believe Alex, mentioned how many of the fields we believe will be, we'll be able to sort of automatically populate. The number of fields that will be able to automatically populate will be higher for studies that are reportable clinical trials that register at clinicaltrials.gov. So if obviously registering at clinicaltrials.gov is already it is a required burden, and that will not go to waste.
Excellent. All right. That is it for our live Q&A. We will now finish off. Can we do screen share? Yes. There we go. All right. Thank you all so much to all the folks who participated in this webinar today. A very special thank you to our presenter, Andrea Tentner, and to our panelists, Alex and Anthony. We are extremely grateful for your insights into HEAL metadata. For the audience, we hope that you're leaving with a better understanding of HEAL metadata, its importance and impact, and hopefully with an increased motivation to share your data among the HEAL community. In addition to a recording of this webinar, we will also email everyone who is registered for this event a list of resources that will assist you as you participate more deeply in the HEAL ecosystem. This slide deck will be shared, and a link to our Slack channel and connect with this forum are located pretty soon in the chat. We hope that you will reach out to us, the HEAL stewards, for any questions or for support as you embark on your FAIR journey to maximize the value of HEAL data. And finally, we look forward to seeing you all at the 3rd annual HEAL PI meeting on April 11th through the 12th, where in addition to a HEAL platform demo, the stewards will also be leading a breakout session to explore collaborations for harmonizing subsets of HEAL data. In addition, in May, we will have our next webinar, so save the date for May 19th from 1 to 2 p.m. Eastern Time. And the topic of this webinar is, if you've heard the term common data elements and you have no idea what it is, or you're familiar with it, but you're not sure exactly how HEAL is going to use these common data elements or CDEs, come and join us for May 19th for that webinar. And with that, everyone, please have a wonderful afternoon. Thank you.